Okay, so that's the electron micrograph of it. You see, it doesn't have the lobulated nuclei, right? So that means it's, it's not a neutrophil or one of the cells that we just discussed, and it's kind of smooth. The cytoplasm doesn't have a lot of granules. So what kind of cell is that? A what? Neutrophil? No. Good guess, though. I mean, a neutrophil is the thing we were just talking about, but it's not the neutrophil. And we're trying to get a very clear distinction in your mind between the cells that have granules and the cells that don't have granules. So this cell doesn't have the granules, and you see it in this electron micrograph, but you also see it over here, looking at a typical stain. You've got erythrocytes, red blood cells. This is just like that blood smear that we did, and you guys saw that there were some cells that were staining. There's two classes of cells. There's the ones with the granules and the ones without the granules. The ones without the granules are called, they're agranulocytes. The ones without granules. Okay, so this one is this is a lymphocyte. So there are two types of agranulocytes. There's lymphocytes and monocytes. So this next picture, that's a monocyte. So again, you have this transmission electron micrograph showing all sorts of incredible detail. And you can actually label little organelles and see lysosomes, and you can see mitochondria and endoplasmic reticulum and all sorts of complexity there. So that's, when you see that, you should know that that's not an optical picture. It's an electron picture. And you see all that kind of detail, much higher resolution. When you see these stains and stuff, that's usually the typical microscope, the compound microscope pictures that we're looking at, and we're using stain. And you're not seeing, it's not as granular. So this is an agranulocyte, and it's a monocyte. So you have lymphocytes and monocytes. So is everybody clear on that? So there's the whole class of cells that are a little bit bigger than red blood cells, than erythrocytes. And if they look grainy, they're granulocytes. And if they look kind of smooth, they're agranulocytes. And then within the granulocyte category, there's neutrophils, basophils, and eosinophils. And if they are smooth, then they're agranulocytes, and they can either be lymphocytes or monocytes. So I'm going over that because you need to know that. So there's some vocabulary that's needed, but it's really worth remembering that. It's going to serve you well to keep that in mind. What's the difference between a leukocyte and a lymphocyte? They both begin with L and they end with a site. And so therefore you lose about half the people all the time on that one. But a leukocyte is another name for what? A white blood cell. It's a big category, a big class. And on one of the lectures, I was drawing these Venn diagrams up here, and I say, well, sometimes, one word is like contained in another word, right? Like, so this is the category of, these are all leukocytes. And there's a subclass of leukocytes that are lymphocytes, completely contained within that, right? So that's really the trick to biology is, knowing not only the vocabulary, but how they relate to each other. And if you get confused on that, you get lost. And then all the other concepts kind of get jumbled up. So it's not so hard to do. So that is required, though, for the class. You understand that, you've got granulocytes, there are three classes of those, and then you have agranulocytes, lymphocytes, monocytes. And monocytes will mature, and we're going to talk about hemopoiesis. They mature into macrophages, which are the major gobbling cells of the body. The next cell here is a platelet. And I think probably half the population couldn't tell you the difference between a platelet and a red blood cell. But we know the difference, right? A platelet is it bigger or smaller? Smaller, right? And it's actually just like a piece of cell. Like it buds off from a much larger cell called a megakaryocyte. And so, and they're kind of injected into the bloodstream. But anyway, so you see here, this is a picture we can relate to. Again, this looks, for our point of reference, this is the erythrocyte. So you guys are going to be looking at a lot of slides, and this is going to be like on the first exam, pictures of blood. And then you see that this is blood because the cells are free in solution, right? And they're kind of round. And when they look donut-like, that's the red blood cell erythrocyte. And then if there's something bigger, that was either a granulocyte or an agranulocyte. And so the ones that are grainy are granulocytes, and those can be either neutrophils, basophils, or eosinophils. If it's not grainy, then it's an agranulocyte, so therefore it's either a lymphocyte or a monocyte. Right? So that level, you guys should all know that. If you see little things, it's probably something smaller than a red blood cell. The red blood cell is kind of the size marker because there's so many of them, and it has such a uniform size. So, and it's about 7.5 microns, micrometers in diameter. But anything smaller than that is probably a platelet. Occasionally there's a large platelet will get released or whatever, but platelets are usually just these smaller pieces. If it's round, it's probably human. If it's elongated, it's a yeast or a bacteria. So, and then platelets are crucial for blood clotting, and then let's look at that. There's a picture of a blood clot. It's pretty cool. So, what's in a blood clot? This is also an electron micrograph. Since you're a histologist, this is really critical too. I mean, I refuse to have people going through histology that can't tell the difference between a micrograph that was made with light and one that's made with electrons. So the ones that are super detailed, like high resolution, this was made with electrons. This is also an electron microscope that took this picture. But it's not a transmission electron microscope, a TEM. It's a surface electron microscope, or an SEM. So this is sometimes called a scanning or surface electron microscope. And what you see here, the round things, these are like cells back in there. It's probably some views of some red blood cells, because that's mostly what's in blood. All these gray things. Maybe we'll do without the lights for now. Kind of a spectacular picture, I think. Very visual. So you see the erythrocytes, red blood cells here, and these are the platelets. And you see that the platelets kind of have a very important role in getting the blood clot to form. And what is the blood clot all about? It's about all of these fibers. It's about fibrinogen being polymerized into fibrin fibers, and that's what the F is here. And so then the P is the platelet, and the rest of the stuff is red blood cells. And we said that's a scanning electron micrograph showing surface features, so SEM. So the transmission electron micrograph, you saw a section. Right? So they're often slicing, I think. And you're looking at the inside of a cell. Which magnification is bigger? The pictures of cells and the TEM, or this picture showing the SEM? What do you think? Because as histologists, that's another important thing. Like, it's all about the picture, and then it's all about, well, the magnification of the picture. How big, how many times was this magnified to see this particular thing? Come on, this is an easy one. What do you think? What? Yeah, TEM is really a high magnification. You can see all of those little organelles inside a cell. Here we're seeing cells, they look pretty small, so that must mean there's much lower magnification. In general, that's true. SEM micrographs are all much lower magnification. You know, similar to what you'd see in an optical microscope. I mean, a little bit higher, but not so hugely higher. But you see so much better detail. Why do you see better detail with any electron microscope as opposed to optical? The resolution is always better with an electron microscope. We don't have one at the minute. We should. I think it was talked about, actually. They put in a grant for an electron microscope. I showed you in the one of the early classes that we have a confocal microscope. And we got that by applying to a medical foundation that granted us that microscope. So it was, you know, $250,000, and they gave us that for free because we were, you know, we're teaching students and we're interested in the next generation of medical and biological, you know, scientists, researchers, doctors. So we could potentially put in a grant for this. Anyway, so the scanning electron microscope, sometimes called the surface electron microscope, it shows lots of detail. So even if the magnification overall is lower than a TEM, it's going to show you all sorts of detail that you just wouldn't see in a regular compound microscope or an inverted microscope. And that's because electrons, that you're beaming this sample with electrons, and the electrons are so small, they're smaller than the wavelength of light. You're limited in resolving power just by the size of light itself, just of its wavelength. Okay. So now I wanted to get into hemopoiesis. At what point do you guys want to do your presentations? I was thinking about 7 minutes tops for talking about your slide. Does everybody have a few things to say about the slide that they chose? I think one way that might be good is it might be a good thing for us to do that now, and then we can return to, I can jump into the hemopoiesis part of it. That way you have a kind of a break. You have to listen to me for a while, and then you get to do something else. Then we can go back to chapter 13, that will be, and we'll talk about how blood cells, the different lineages mature. How they start out as one thing and they take on these other characteristics. Does anybody need to use a computer? Does anybody have a file to show or a website that might have something online to show? This is kind of practice. I think it's okay that you know, try to feel relaxed about it, and just like Connor was up here and he drew the picture of a blood vessel microcapillary, and you know, saying, well, the neutrophil is going to squirt through. Just kind of, you know, come up. You could use some pens and say, well, you know, I had this slide and it was about a thalassemia, and that's this kind of thing. And you can start to draw some red blood cells and say, well, what's different than thalassemia? What would you see? Does that sound okay? Okay.